Hello, everyone. My name is Merlin Herrera.
And my name is Ratemo Vincent.
We will be discussing the paper called Melittin Triggers Apoptosis in Candida albicans through the Reactive Oxygen Species-Mediated Mitochondrial Caspase-Dependent Pathway. Now, melittin, this is a poison, a toxin that is found in the poisonous honeybee, Apis mellifera, and this bee prefers warm weather with lots of vegetation, lots of tree around. This bee is native to Europe. In figure 1, we have included a visual image of melittin. As we can see, it's not very organized, but this image gives us an idea of what melittin looks like. And now we should know that melittin is a fairly toxic compound because, according to research, very small amounts are required to cause changes in the plasma membrane of Candida albicans. Nanomolar and micromolar amounts of melittin are just enough to cause pore and bacterial membranes, as we will see in this paper.
So in this experiment, we used HPF staining method to detect melittin-induced hydroxide, and what they found out from the results is that melittin induced the cells that were stained with HPF. Cells that were stained with HPF induced higher hydroxide generation. That, according to the graph, showed 76.12%. That was more than the negative control percentage. And when looking at the cells that had hydrogen peroxide, there was it showed a significant higher percentage of 97.27%, and demonstrated more potent inductive activity. So the presence of diarrhea, that's the hydroxide scavenger, controls a significant portion of melittin-induced reactive oxygen species.
In the next figure, what we look at is a JC1 staining assay, which was used to detect the changes in the mitochondrial membrane potential, as shown in this figure. What the researchers did was that they created 3 groups of samples of Candida albicans. One of the groups was a control group, which did not have any substances added to it. In the next group, they included a sample of Candida albicans, which included a sample of melittin, and the third group was a sample of Candida albicans cells, which was applied a certain amount of H2O2. And what they observed was that in normal cells, there were delocalized positive charges observed as red fluorescence. In this image, unfortunately, the red fluorescence was not included, but that is what the researchers found. In apoptotic cells, the dye remains in the cytoplasm as green fluorescence dye. Again, the dyes were not included in this research paper, and that is one fault that we found with this paper. The ratios of y-axis to x-axis changed from 5.1 to 0.6 in melittin-treated cells, which indicates that there were changes in the mitochondrial membrane permeability. And as this paper discusses, the application of melittin to cells tends to cause changes in the permeability, allowing more substances to go in and out of the cells.
So in the next slide, it shows the fluorescent microscopy of Fura 2AM and Rogue 2AM. Those are 2 dyes that detect the cytosolic calcium concentration and mitochondrial calcium concentration, as seen in the diagram. The 2 dyes show that melittin caused the melittin and the Pura 2AM caused slight increase in intracellular calcium levels in Pura 2AM, and and this showed that it had remarkable increase in intracellular calcium levels. That was the road to So based on the figure, it indicated that the mitochondria are involved in apoptotic pathway of melting in Candida albicans.
In the next figure, we look at the experiment in which Candida albicans cells were treated with Melittin and H2O2 to observe any metacaspase activation. In the first part of this image, in part A, we observed that they analyzed Melittin-treated cells and H2O2-treated cells. In the control group, the Candida albicans cells weren't given any Melittin or H2O2, and the other two groups that they considered were Melittin-treated cells and H2O2 cells. Melittin-exposed cells showed an increase in fluorescence, which indicates intensity of metacaspase, and that is seen in figures A to D. Indication of metacaspase inducing Melittin fungal cell apoptosis, which means that the higher the intensity of fluorescence, we can say that there is a higher indication of apoptosis. Cytochrome C concentration in mitochondria decreased after being exposed to Melittin. And also, the cytochrome C concentration increased in the cytosol. And from this figure, we can conclude that cytochrome C is a marker of apoptosis because as apoptosis occurs, cytochrome C is released in higher and higher amounts.
So in conclusion, what's seen from the paper is that the Melittin-induced apoptosis in Candida albicans is triggered by hydroxyl radial generation, mitochondrial membrane permeabilization, and metacaspase activation. Melittin, as seen in the paper, disrupts the fungal cell membrane of Candida albicans with potent activity through the apoptotic mechanism pathway. So based on the research study, it suggests that Melittin could trigger fungal apoptosis after membrane damage using a dual mechanism against pathogenic microorganisms. However, according to research, they still haven't been able to figure out how these two mechanisms will do that, and so more intensive studies and investigations need to be done to come to a conclusive hypothesis.
And we must add that in this research paper, it was concluded that metacaspase was a central regulator of apoptosis, and they are trying to conduct experiments which prove or disprove this. The results that they found also indicated that metacaspase activities is correlated with mitochondrial membrane permeability, but it's still an ongoing research. Thank you. Sorry, the references.